Hello students, welcome to today's lesson on transfer of nutrients, the second part of transfer of nutrients in an ecosystem. In this lesson, we are going to study nitrogen cycle in an ecosystem. Nitrogen is an essential component of genetic material DNA and RNA and the principal structural materials or proteins, the principal energy transfer molecule ATP, and all organisms require nitrogen to live and grow. Nitrogen is required in substantial amounts by all living organisms. In many ecosystems, nitrogen is the chemical element in shortest supply relative to the needs of organisms. A paradox is that the atmosphere is supposed to contain 78% of nitrogen by volume, but the vast majority of organisms, including all plants and animals, have no way to use nitrogen from its diatomic molecule form N2. This problem occurs because most plants can only take up nitrogen in two solid forms, ammonium forms and nitrate forms. Most plants obtain the nitrogen they need as inorganic nitrate from the soil in solution forms. Ammonium is used less by plants for uptake because in large concentrations it is extremely toxic for plants. Animals receive the required nitrogen they need for metabolism, growth, and reproduction by the consumption of living or dead organic matter containing molecules composed partially of nitrogen. There are three types of soil bacteria that play an essential role in supplying plants with nitrogen. These three groups of soil bacteria are nitrogen fixing bacteria, ammonifying bacteria, and nitrifying bacteria. For animals, the ultimate source of nitrogen is nitrogen containing organic compounds synthesized by plants, algae, or other microbes. The main processes of nitrogen cycle. Nitrogen cycle involves the following major processes. The first one is nitrogen fixation. This nitrogen fixation is the process by which atmospheric nitrogen is converted to nitrogen containing compounds. This can be done mostly by biological fixation. This biological fixation involves living organisms. In this case, atmospheric nitrogen is split and combined with hydrogen atoms to form ammonia by some free living bacteria in the soil, like the genus Azotobacter and Klebsiella, and by symbiotic bacteria that can form a symbiotic relationship with leguminous plants. In this relationship, rhizobium bacteria colonize plant roots, causing the formation of nodules. From inside these nodules, the bacteria absorb nitrogen gas from the atmosphere and then transform it into nitrate or ammonia that are usable by bacteria itself and the host plant. In exchange for the nitrogen, the symbiotic host plant feeds the bacteria with carbohydrate that it produces from photosynthesis. The second way of nitrogen fixation is high energy fixation. In this fixation, a small amount of atmospheric nitrogen is fixed by lightning. The high energy combines nitrogen with water, resulting in ammonia and nitrates. These forms are carried to Earth in precipitation. The third way of nitrogen fixation is industrial fixation. In this process, under a great pressure, at a temperature, and with the use of catalysts, atmospheric nitrogen and hydrogen, usually derived from natural gas or petroleum, can be combined to form ammonia. Ammonia can be used directly as fertilizer, but most of it is further processed to urea and ammonium nitrate. Human use of nitrates in fertilizer has doubled the available nitrogen to plants. Then plants can absorb nitrates from the soil to make plant proteins. The second process of nitrogen cycle is ammonification. Ammonification is decomposition or decay with production of ammonia or ammonium compounds, especially by the action of bacteria on nitrogenous organic matter. The process of ammonification is the result of the breakdown of organic matter such as dead animals and plants or waste materials like excrement. This breakdown is accomplished by microorganisms which utilize dead organic matter for their energy and produce ammonia as byproducts of their metabolism. That means decomposers like bacteria, fungi, and some protozoa decay excretory products and detritus to release ammonium ions from nitrogen containing organic compounds of the dead body. Ammonifying bacteria here is a good agent to break open dead bodies of organisms to release nitrogen in the form of ammonia. The third process of nitrogen cycle is nitrification. Nitrification is the production of nitrates from ammonium or ammonia. This process is accomplished into two steps. One is bacteria of the genus Nitrosomonas oxidizes ammonia to nitrites. The second one, bacteria of the genus Nitrobacter just oxidize nitrites to nitrates. These two groups of autotrophic bacteria are called nitrifying bacteria. The fourth process of nitrogen cycle is denitrification. Denitrification is the process of converting nitrites and nitrates back into atmospheric nitrogen gas. This replenishing or fill up again the atmosphere. Once again, bacteria, which is called pseudomonas, are the agents to convert nitrates or nitrites back to nitrogen gas. These pseudomonas live deep in the soil and in aquatic sediments where conditions are anaerobic. These bacteria use nitrate instead of oxygen for the final electron acceptor in their respiration. So this denitrification just counterbalances nitrogen fixation with some extent. As we can see from the diagram, nitrogen recycle between the living system and abiotic environment. As we said before, most of the nitrogen accumulate highly in the atmosphere. That is, about 78% of nitrogen gas gets accumulated in the atmosphere. In this case, there are responsible groups of microorganisms that can fix nitrogen to ammonium. Nitrogen fixing bacteria just fix nitrogen gas into ammonium ions in two main situations. One, nitrogen fixing bacteria free in the soil, Azotobacter and Klebsiella, as we can see from the diagram, capable to convert this nitrogen gas into ammonium ions in the soil. The second groups of bacteria, nitrogen fixing bacteria in root nodules, as you can see, that belongs to the genus Rhizobium, form ammonium ions or nitrates that are passed to the leguminous plants and used by them to synthesize amino acids and hence proteins. Since all living organisms are mortal, they can die after they accomplished their lives or existence on Earth, and they can also excrete nitrogen containing organic compounds. Then ammonifying bacteria just decompose or break open this detritus and release nitrogen in the form of ammonia to the soil. And then nitrifying bacteria, that is Nitrosomonas and Nitrobacter, just converts ammonia to nitrites and hence nitrates respectively. When this nitrogen gets accessed into the soil in the form of nitrates, plants can absorb these nitrates to make plant proteins. This nitrogen then pass through trophic levels to primary, secondary, and tertiary consumers. There is also another group of bacteria, what we call pseudomonas, that capable or specialized to convert nitrates or nitrites back to nitrogen gas to the atmosphere. This is the images of root nodules in which symbiotically related bacteria and leguminous plants can form. At the moment, researchers have conducted an immense research on the genes of nitrogen-fixing bacteria. Scientists just aimed to isolate these genes that can control nitrogen fixation, and they wanted to transfer these genes by genetic engineering into other plant cells in order to increase productivity of these plants. The second aim of scientists is pursuing bacteria like rhizobium to form symbiotic relationship with other species of crop plants. If all the cereal plants that are grown in the world had nitrogen fixing bacteria in their root nodules, or if their own cells could fix nitrogen, crop yields in countries with extreme food shortage would be hugely increased, and the plants would not need nitrogen fertilizers. It saves money and energy expenditures to make and have fertilizers, nitrogen containing fertilizers.